substrate number one. And it's an illustration of how we can activate a number of different pathways through different mechanisms in regards to how this insulin substrate number one interacts with its various associated proteins. In this case, when we look at the extracellular regulatory kinases that promote potentially proteins expression in the nucleus, and some of these particular proteins are going to be those enzymes that are involved in glycolysis as well as in gluconeogenesis. Again, it's an illustration of a balance in regards to if we're increasing the glucose level, we are going to have to bring that glucose in, which we'll see in a second as we saw at the end of yesterday's lecture. But we're going to have to do something with that glucose that's come into the cell. And we're going to have to either metabolize it or we're going to have to store it. Now, if we're going through the metabolistic approach, we're going to have to mostly increase the number of enzymes that are present to ensure metabolism in regards to consuming this glucose. So this is where we see the insulin receptor insulin substrate number one playing a role in regards to forming a protein complex. And we've already discussed what this protein complex does in the form of phosphorylation to basically phosphorylate this protein, the extracellular regulated kinase. And once it's phosphorylated, it can move through selectively through the membrane of the nucleus and then promote a phosphorylation event to increase the production of particular proteins. And as I said, in these cases, most probably those to do with glycolysis and gluconeogenesis. But when we look at also the insulin receptor substrate number one, we also have it working at another system where basically it's now increasing the concentration of the glucose as a result of the insulin receptor being stimulated and phosphorylating the insulin receptor substrate number one. This one works on a system we're already aware of, which is the IP3 receptor, and then activating the protein kinase B. So go back to your IP3 receptor, and you'll see how we see phosphorylation working with IP3 to get that coordination and the activation of the protein kinase B. Now, with the protein kinase B, this facilitates, importantly, the movement of the vesicles that contain the transporters, the GLUT4 transporters, to move to the membrane. And with that membrane annealing and the receptors being, or the transporters being in the right orientation, we then see more movement of glucose coming in. But that means, again, we are going to, one, promote the genes and proteins that are going to consume that glucose. But if our metabolic profile is different, we might want to be able to store that glucose instead of metabolizing it. And this is where we basically see now the protein kinase B playing a very important role in phosphorylation of a substrate called the glycogen synthase kinase number 3. Now, this is a key enzyme in regards to whether you're taking glucose and making glycogen, which is the storage element of glucose. In this case, when the glycogen synthase kinase number 3 is active, it's active in its dephosphorylated form. And what it does, it basically binds to the glycogen synthase, which is the actual enzyme that takes glucose to produce glycogen, which we'll study a little later on. And it basically puts a phosphorylational group on there. So once that is phosphorylated, there is no glycogen synthesis. So what I'm trying to illustrate here, there's a menagerie of interactions. Phosphorylation can be both positive and negative depending on the enzyme. And in this case, we have enzymes working with enzymes to regulate the production or degradation of certain things. In this case, the production of glucose, production of glycogen, which is matching the transportation of glucose into the cell. So in this case, when we have the glycogen synthase kinase number 3 in its dephosphorylated form, it's active. But the protein kinase B has the capacity to take this particular enzyme, phosphorylate it, causing a three-dimensional structural change in the glycogen synthase kinase number 3 to then cause its dissociation and basically now allow the glycogen synthase to become active, consuming the glucose that's entering in the cell by the GLUT4 transporters, and then produce glycogen. So basically, it's a very complex system where basically phosphorylation causes three-dimensional structural change with these proteins and allows then their regulatory components to play a role in whether they're switching enzymes off or enzymes on for a particular goal. But the important thing is everything is matched in regards to if you're bringing glucose in, you're going to have to do something with it. You're either going to metabolize it or you're going to actually store it. Okay, so again, we have to be considerate of what's coming into the cell and what are the needs of the cell. And we'll talk more about that in coming lectures. So our next example is a protein kinase, which basically is a tyrosine kinase. And this is similar to what we saw a little bit with the fibroblast systems where you have dimerization occurring. And we need this to occur first off to allow a process of regulation. And that regulational process requires then a ligand to bind two soluble proteins to become a dimer. And this particular tyrosine kinase is important in red blood cell formation. It basically utilizes an erythroprotein, you might have heard it as EPO, and this particular protein is a plasma membrane protein and basically triggers off the whole complex by forming a dimerization. And once we have this soluble protein binding, we then produce what we call a Janus molecule. Now, Janus is a Greek, I think it's actually a Greek or Roman term. I think it's a Roman term. It means two-faced. So there's a face facing forward and a face facing backwards. So they use it like in their households to keep away evil spirits that people come and go. They're always seeing the front and the end. So they call them JAK molecules or Janus kinases. Now, the JAK phosphorylates the tyrosine in the kinase, particularly that of the cytoplasmic side of the EPO receptor. We'll see a picture of this in a second. And that then actually coordinates a whole process of signal transduction through dimerization, and then importantly with that dimerization, allowing a signal to actually get to the nucleus, and again expressing specific enzymes, proteins, to produce, in this case, red blood cell functionality or red blood cells themselves. But again, note the promiscuity of the cascades, where there's interface with other cascades like the MAP cascade, which can then interfere with what we call the GERB2 protein. So that GERB2 protein is an essential part of that particular MAPK cascade and can interfere with that. And they're also interfering with the process of gene regulation by stimulating a regulator to go through the nucleus membrane. So let's have a look. So here's our dimer of the EPO receptor, the erythroprotein receptor, and it's usually dissociated in the membrane, and it will only come together when erythroprotein is present as the ligand. Once that's actually occurred, there's a process of phosphorylation and interactions with various other pathways, as we've seen with the MAP2 pathway, promoting the process in regards to the MAPK affecting gene expression. So we've already seen that. But the other thing, what happens is these nuclear ligand stimulating domains, which basically allow these particular STAT molecules through their process of phosphorylation to form a dimerization by interacting with each other. So they're the same molecule, but due to their phosphorylation in this situation, they interact with this SH2 domains and form this dimer component. Once they form that dimer component, they have these nuclear localizational sequences that bring in them into the nucleus through the membrane and also then bind to the DNA to increase messenger RNA and allow impact on gene expression. So again, we have this capacity to interface different pathways. And in this case, the focus is dimerization through a regulatory ligand on a receptor in a membrane to then cause dimerization of a molecule that is going to activate gene expression. So in this case, we have a number of different systems increasing gene expression, gene regulation, because there's a response that is needed to be met. And again, it's a balancing situation. And there's also going to be causing dissociation of this particular component. So once the erythroprotein is dissociated from the complex, the EPO receptor dissociates as a dimer form, and therefore we inhibit any of the MAPK cascade and anything regarding the nuclear localizational sequences in the SAF molecules and forming that particular dimer, and then promoting the issue of gene regulation. So we have this capacity to switch things on and off, but the idea is there are many pathways that are integrated to do the same thing or similar things. And in some cases, there's a heavy reliance on one thing and the other to actually get the job done. So let's have a look. So another one we have is the guanylyl cyclases. Now, this is known as a receptor enzyme, and it produces an agent called cyclic GMP. Now, we haven't come to cyclic GMP, we've come to AMP, ATP, and ADP, but we're going to look at cyclic GMP as a secondary messenger. So here is the nucleoside in regards to the guanidine triphosphate, and it causes the release of the inorganic pyrophosphate. So two phosphate groups come off. And with that remaining phosphate group in the number 5 position, remember our carbohydrate chemistry, and the 3 position of the hydroxyl group, we actually produce a phosphodiester bond in regards to utilizing this hydroxyl group and this phosphate group, causing a cyclization between the 3 and the 5. Now, this is really important because this now becomes a very highly active transmitter in regards to binding to particular tissues and basically activating those particular tissues. So in the kidneys and the intestines, the production of cyclic GMP can change various water absorptions as well as the retention of certain ions, particularly salts. And this usually gets screwed up when you have diarrhea in regards to you're not absorbing water. And one of the big things, particularly in diseases such as cholera, is actually the loss of sodium chloride and water, which can be very, very, very detrimental and even fatal. In cardiac tissues, the cyclic GMP causes muscle relaxation. So there's a lot of interest in that, in basically decreasing, decreasing blood pressure. In the brain, it is a long-term constituent for brain development. So we need a constant supply of this in our juvenile states to allow us to actually mature and actually develop these various parts of the brain. Now, the guanylyl cyclases are activated in this case by an atrial natriuretic peptide. And this is basically in response to what we see in high blood pressure. As I said to you before, there's a very interesting component in regards to muscle relaxation and basically causing this situation to basically be expressed when we see an increase in blood pressure. Because what we want to do in the system, and this is how the body naturally works with high levels of blood pressure, is to basically work out what we do with the blood volume. So think of it this way: if you can decrease the blood volume, you can decrease the pressure in the system. Okay, so what we see is with the cyclic AMP and the activation of the atrial natriuretic peptide, we see an activation in regards to secretion of sodium chloride, particularly sodium salts or sodiums. And in this case, we see an increase in their excretion. And as we know through osmolarity, when we move a sodium ion across a semipermeable membrane, there's going to be a difference of that particular salt, which will then carry water with it. So that increases the loss of water. In that case, we actually decrease the blood volume, and therefore we cause then vasoconstriction, vasodilation, not constriction, vasodilation, relaxation of the blood vessels. And the overall outcome is decreasing the blood flow and decreasing the blood pressure, all because of this secretion of this little molecule. Now, the interesting thing is this molecule has a very, very short lifespan. There are other enzymes that want to break it down very quickly, because if we have this stimulating too much, we can cause some very dramatic situations in regards to fainting. And some people have problems in regards to regulating their own blood pressure because they may not be secreting or producing enough of this cyclic GMP. And you can read more about it on page 466. But this particular molecule is made by an enzyme, okay, a membrane-bound enzyme. Now, this gets to what I want to focus on today, and is usually presented in the homework and in the exams, are these G-coupled protein receptors and secondary messengers. And basically, these are known as GCPRs, and they are a very highly pronounced target for drug design and development, and play a very essential role in establishing homeostasis within our biological systems. There are three essential components that make up these GCPRs, or G-coupled protein receptors. That's what GCPR stands for. And there is a plasma membrane receptor, and it's usually 7 transmembrane helical segments. There is an enzyme that's associated with this plasma membrane, and that particular enzyme, like we saw with the GMP, produces a secondary messenger. And then we have another protein which basically binds that secondary messenger and does a function. Okay, so there's 3 parts there, and that's the guanosine nucleotide binding protein, or actually what we call the G protein. So we have a plasma membrane, an enzyme that produces a secondary messenger system, and a protein that binds that secondary messenger system and allows a particular function to occur, usually a process of phosphorylation. So what we see is an activation of this receptor in one way, shape, or form by an external stimulus. And that G-coupled protein, basically the G protein, exchanges the GDP to GTP. So it goes through a process of phosphorylation from the di to the tri. That GTP is still attached to the protein. So that becomes then basically the GDP is attached to the protein. Once it becomes phosphorylated, it then causes an dissociation. So that GTP then dissociates from that protein complex where the phosphorylation occurs, has the solubility to move across in a very local area and bind a nearby enzyme. And upon that particular binding, it then impacts the enzyme activity. We're going to see this in a picture in a couple of slides. I'm going to focus on the beta-adreneric receptor, because this is an example of a G-coupled protein utilizing a compound that we're a little familiar with in the form of cyclic AMP. So a very similar version to what we saw with the cyclic GMP. And this guy is basically going to be a very important modulator, as we'll see a little later on, in regards to regulation of the metabolism of certain components within our bodies, particularly that of carbohydrates and also fats. So we have 4 different types of beta-adreneric receptors. They're called the A1, A2, B1, B2. And the interesting thing is they do a similar function in regards to producing cyclic AMP, but they are found in different forms in different tissues to give you different capacities of regulation and control of these beta adrenergic receptors. And their responses also may vary in regards to what occurs through the stimulational process and the resulting activity in the process of phosphorylation of a particular enzyme creating a particular event. Meaning we have these capacities to have separate but equal systems, allowing us to modulate and control them using a similar same system, but the end point is going to be different. So keep that in mind. So let's focus on the beta adrenergic receptor, which basically is this system, as we'll learn a little later on, in metabolism, which increases both the breakdown of glycogen and fats to provide us the energy constituents to consume to produce ATP and other energy requirements, to meet other energy requirements. So we have this integral protein, the 7 membrane, transmembrane spanning regions, and they're usually called 7 TMs because they are 7 transmembrane spanning components. And they snake across. They're a single amino acid sequence, a primary sequence of 7 transmembrane domains, and they cross the bilayer. This is why they're called serpentine receptors, because they snake across the lipid membrane. Now, when we look specifically, and this is the wordage we're going to have on the extras on the next slide, is think about this. With our epinephrine or adrenaline, so there's two terms we can use interchangeably, and I use both of them interface, meaning the same. So epinephrine and adrenaline, basically when these molecules are released through the fight or flight system, so you've had a big scare, and we're going to see the epinephrine and adrenaline basically being released in the circulatory system, and a whole cascade of things occurring in regards to making the body ready to flee. That body, regarding the muscle contraction, is going to need enormous amounts of energy. And this is where basically we get this conformational change to basically promote a rapid response to causing the release of energy, either from carbohydrates, from our glycogen, or from our fat stores. So that's going to be talk about that a little later on. So I don't want to put it in the right framework. But when we see the binding of these particular stimulatory compounds, they cause a three-dimensional structural change. And with that three-dimensional structural change, like we've talked about so many times in a protein structure, we're going to get some type of effect occurring. In this case, we get an interaction with a secondary protein that is in the cytosol of the cell. And that is what we call the GS, or the GTP, binding symmetry G protein. This is the guy that stimulates, importantly, the production of the cyclic AMP by another enzyme. So again, it follows what we've talked previously in regards to the 3 components that are required for GCPRs. So I'm going to go through the step-by-step component, and then I'll show you the picture, and we'll go through it again. So this is the importance of these GCPRs. So we basically have this stimulatory protein being activated by the norepinephrine and adrenaline, and basically taking the GDP and binding GTP to the activation of the adenyl cyclase. But in the same process, when we have that phosphorylation event occurring, we have the GDP being bound and inactivating it. So the activation of the stimulatory protein is based on a single event of phosphorylation with the bound nucleotide. So in this case, when we have the stimulation of the external source, in this case with an epinephrine or adrenaline, we are seeing that GDP being displaced. Therefore, the stimulatory protein is then activated or reactivated. This particular dissociational protein then basically interacts with a alpha class of G proteins. And this now becomes a stimulatory protein with the alpha class stimulational component, because what it has now is that protein is now bound to GTP. So remember what we said with GTP, the GS is activated. With this activation, we have a solubilization, which then moves this particular activated protein to the adenyl cyclase. Once that adenyl cyclase is interacted with the association of the GS alpha with the adenyl cyclase, we start then seeing an increase in cyclic AMP because the adenyl cyclase is there to consume ADP or ATP. In this case, it would be ATP. I should make it clear. It's ATP to produce cyclic AMP. Cyclic AMP concentrations then increase in the cytosol because of this activation of this enzyme. But we now need to turn this off because if we get too much stimulation, we're going to have too much cyclic AMP, which is going to cause a lot of problems. Because remember, cyclic AMP is what we call a localized hormone, okay, a localized transmitter. And we need to be able to control this because it can have high levels of systemic issues because it's also a little promiscuous in regards to what it interacts with. So as we see with the GS alpha, we can have a GTPase, which then can basically cut the phosphate group off the activated GTP in the stimulatory protein and bring it back to the GDP, which is in this case inactivator. And with this inactivation, we get dissociation of the stimulatory protein from the adenyl cyclase. We stop the reduction of cyclic AMP, and therefore this stimulatory protein goes back to its original complex and associates again with the beta and gamma subunits of the protein of the receptor, and basically the cycle then can start again. So it's more illustrated in what we see, and you're going to have to know this in regards to the next slide. Hopefully. There. Okay. So let's go on and have a think about this to what I've said before. So we have a number of cytosolic subunits that are attached to the lipid membrane and are associated with a transmembrane protein. And this is our serpentine receptor. And in this case, we could have different types of receptors. We could have a vasopressin receptor that binds a peptide to cause the same biological effects in the sense of phosphorylation and cyclic AMP production. We can have a whole load of different receptors here, but this inside mechanism here, is basically the same. So in this case, we have binding of the beta-adreneric receptor, and here's our subunit components. In this case, the cytosolic component is pre-armed with the guanidine diphosphate, or GDP, already. Now, what we see is a process of exchange of that GTP in the process of phosphorylation, where this guy now is removed, so this guy is open, and therefore basically becomes activated with now the binding of the GTP. With that GTP, we then get this GS alpha being dissociated from the protein complex. These other remaining parts, the gamma and the beta subunit, are still together with the receptor because they're bound by the lipid membrane and are bound to associated to the beta-adreneric receptor. But with this GTP being activated and bound to the alpha S stimulatory unit, which is now activated, it has the capacity to move along the inner leaflet of the membrane because it's attached to a lipid in the inner leaflet of the membrane, and has the capacity to move across. And it'll only do this because of that extra phosphorylation event causing its dissociation from the beta and the gamma subunit. With this phosphorylation event occurring, it then has the capacity to bind to another transmembrane protein, in this case the adenylyl cyclase, which has the capacity to consume ATP and produce cyclic AMP. This only occurs with the stimulation of the loaded GTP stimulatory alpha S protein. With that in mind, we then have the capacity to consume ATP to produce then AMP, in the sense of cyclic AMP. Now, with this particular cyclic AMP now being produced, we can interact with protein kinase A, which is going to go through a number of phosphorylation events in regards to the epinephrine stimulation. That protein kinase A can increase the mobilization of enzymes to cause degradation of glycogen and of fats, to give us that energy boost. It can also be utilized for constricting blood flow, to increasing the capacity of movement of materials through the thing. So anything you think about when you get a good scare is because of that cyclic AMP activation. But as I said, the issue is we also have to degrade this, and it's very important we degrade this. And we degrade this using a cyclic nucleotide phosphodiesterase. And it cuts that phosphodiester bond to produce then 5' cyclic AMP, which then basically can be utilized as a degraded component to prohibit potentially as a competitive inhibitor of the cyclic AMP activation of the protein kinase A. So you're going to have to understand how this works in regards to the individual steps. But when we see the deactivation, I think my next page is my next slide. Let me just check. Okay. So we're going to talk a little bit later about, in the next couple of slides, about its deactivation, because the deactivation is the reverse, what we'll see in regards to this important GTP being basically primed now to become ADP, causing its association with the adenyl cyclase, and then migrating back to the beta alpha subunit. And again, repriming the system with the beta adrenergic receptor to then accept more epinephrine to then cause that stimulation to occur. Now, the issue with this cyclic AMP and this 5-AMP becomes very, very important. Let's have a look at the structures of these particular molecules. So we have ATP. So we now understand now ATP is not just an energy source. It's actually now a precursor for a number of transmitting events which are going to increase activation of enzymes and cause maybe the increase of enzyme secretion by interacting with the proteins within the nucleus to increase the process of protein expression. Now, the issue of this particular guy is we're going to see what we saw exactly the same with the GTP, is the removal of those two phosphate groups, breaking of the phosphoanhydride bond, and giving us PPI, the inorganic phosphate, the inorganic pyrophosphate, I should say. And that is using an enzyme called the adenyl cyclase, which we illustrated in the previous slide. The structure itself, knowing our carbohydrate chemistry, we see the 5' and the 3'. So we still call this adenosine 3, 5' cyclomonophosphate, or in this case, cyclic AMP. Now, this again is a highly reactive constituent that has a little bit of promiscuous activity. So we don't want to make lots of it and where it can diffuse away from the target enzyme. So what we want to do is make sure it's short-lived, and we do that by using a cyclic nucleotide phosphodiesterase, utilizing molecular water, again, molecular water becoming a part of the reactional process, and basically causing the cyclic component here to actually be cleaved in the phosphate 3 position, this here. So we then basically have the capacity to put the hydrogen group here, an oxygen, and then put another oxygen on the phosphate, and producing then the monophosphate version, in this case the adenosine 5 monophosphate. So indicating 5 indicates where the phosphate group is. It's not left on the 3, it's on the 5. So this particular guy can then be recycled, later on uptaken, be phosphorylated if need be, or participate in other reactions as a controlling regulator. And we're going to see this guy, both AMP and cyclic AMP, playing a role in which way we're going, whether we need to undertake fatty acid metabolism and glycogen metabolism to provide us those energy sources, because the cell has been given a stimulus. So again, we're going to have this seesaw balance in regards to what molecules are around, what molecules are going to affect particular needs of the cell in define of response. And it could be low glucose level, it could be the secretion of epinephrine and adrenaline. So we're now looking at the issue of the inactivation of this particular receptor. And we're going to go through a number of little processes to go through the dephosphorylation. So we already understand how we become the active form with the adenyl cyclase from the, in this case, the beta adrenergic receptor with that phosphorylational displacement of the GTP and replacing it with a displacement of the GDP and replacing it with a GTP. Once we get that phosphorylation again, three-dimensional structural change in the alpha subunit, moves across the adenyl cyclase. Now, what happens here is interesting because it causes this dissociation to become completely cytosolic soluble. In this case, it's a dissociation of the alpha subunit is caused by dephosphorylation of the GTP that is bound to the active constituent. And with that comes a phosphorylation or a dephosphorylation event, which basically now allows the adenyl cyclase to dissociate itself from the alpha subunit. With that complete association, conformational structural change that occurs through that simple dephosphorylation of one phosphate group, we then get complete solubilization and the high affinity interaction. So these guys have an interactive surface that has a high affinity association with the beta and gamma subunit. So it's now primed with GDP. It's going to be no longer activating the adenyl cyclase, and then basically has this capacity now to interact with the remaining parts of the subunits, which are now inactive, to then cause an activation of a new complex, the same complex that we started with, and it's being primed already with the GDP to go through the GCPR system and go through the whole process again. So we're going to have to also think about the inactivation of this particular system. So here's the three-dimensional structure of the adenyl cyclase, and the structure of, importantly, the GS alpha protein. And you can see there is a high homology in regards to a near perfect kind of associational fit. And this only occurs when this is the molecule, this is GTP sitting here, is present. When we see the removal of that individual phosphate group, we get a changing of the interaction here due to the destabilization of this structure. It comes a little bit open more, and therefore causes a three-dimensional protein change in the binding region or the association interface between the adenyl cyclase and the activated GS alpha protein. And with that, when we get that GTP going to be dephosphorylated, we can understand now how it dissociates from the adenyl cyclase. So again, we go back to our protein structure, function, and form, and how now just a simple change of a phosphorylation event on a protein, which is, in this case, it's a nucleotide binding region. So we have a nucleotide binding region. That phosphate group plays a particular role in providing those negative charges, maybe pushing positive charges to cause a little kind of a change in the three-dimensional structure. But without that additional negative charges, that three-dimensional structure then returns back and then becomes an inactivated interface and no longer has interaction with the protein of interest. So again, I keep going back to the issue of three-dimensional protein structure and how events are changed due to these simple phosphorylation events in regards to bringing in those negative charges. And as we now know, those negative charges are going to have interface interactions with three-dimensional structural elements within a structure and cause its three-dimensional change and then cause its dissociation. So hopefully this message is getting through to you in regards to these dramatic changes that we see in protein structure. So I'm now going to illustrate here what is the situation with now the production of that cyclic AMP. That cyclic AMP is being produced locally, and it's increasing its concentration in local regions in the cytosol. Why do we want this cyclic AMP to actually be there? Because what that cyclic AMP does, it acts as a regulator to the protein kinase A. So here's the protein kinase A being bound with various elements of the cyclic AMP. But noticing, importantly, there is multiple binding sites of the cyclic AMP for this particular protein kinase A. So protein kinase A is a cyclic AMP dependent protein kinase, and it's specifically acted by a number of cyclic AMPs. Now, the reason we have a number of cyclic AMPs is because if we do not reach the required number of binding of cyclic AMP, the protein doesn't work. So we have 4 binding sites in regards to binding cyclic AMP. So we need to produce 4 amounts of cyclic AMP to activate one protein kinase. Because again, we're working on a system where we're producing an enormous amount of cyclic AMP. We want to activate the protein kinase. But if we're only doing like a half measure, we may not want to activate it as much as the protein kinase. So the protein kinase is sitting there waiting to be activated, dependent on the cyclic AMP production. So depending on how much cyclic AMP we produce is going to depend on how much now response we're going to get from the protein kinase A, because we can activate a number of those, given that we need 4 requirements of cyclic AMP to bind them. So let's have a look. So what we see is a regulatory and catalytic site of the cyclic AMP, of the protein kinase A, awaiting the cyclic AMP binding. In this case, when the cyclic AMP is not bound, this protein is inactivated. Once we get activation occurring where 4 moieties of cyclic AMP are binding to the regulatory components here, 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 and here, we see a dissociation of this high affinity protein complex. In its uncyclic bound situation, it has a high affinity to the other part of the protein, to the catalytic sites. But when that cyclic AMP binds to the regulatory component, 3-dimensional structural change occurs, basically causing then it's a dissociation and then opening up a cleft of the enzyme, the catalytic site in the protein kinase A, then to do its particular job. But if we remove the cyclic AMP from the regulatory component, and because of its high affinity between the 2 protein structures, we remove the cyclic AMP from the regulatory component, it's going to have a high affinity to bind back to the protein kinase A, and then prohibiting its biological activity. So again, it's an illustration now of protein conformational changes that occur through regulatory constituents and basically open and closed catalytic sites. So here we have the regulatory subunits with a process we call auto-inhibition. So we have this capacity in regards to these domains being buried within the structure of the regulatory component, causing its dissociation. And then the catalytic sites then become open to the new substrates. And again, those substrates could be then phosphorylated because it's a kinase, it's phosphorylating something. And those substrates then be phosphorylated, do something else, or actually become something in regards to a down chain event in increasing protein transcription, changing blood flow, increasing enzymatic activity. So you can see the point I'm making here, that protein kinase A is a portal to a lot of other biological events. But that means it must be very closely and guarded and regulated. And we regulate that by adding these additional binding sites, as well as the conformational high affinity of the regulatory subunit to cause dimerization and trimerization, or tetra 4 subunits dimerizing, in regards to the deactivation of the protein kinase A. And we're going to come back to protein kinase A much later on when we look at metabolism, and we're going to bring back that issue of cyclic AMP playing a regulatory role. So again, an introduction to what's occurring in this particular system. Now we know this is happening because we can do some very snazzy work in regards to these particular proteins. We understand there are a regulatory component, catalytic events, and this particular dimerization or cleavage site, which occurs when a protein interacts and changes its interaction with its catalytic site. So in this case, again, similar to what we saw with the other structure, it's the same kind of thing but a different orientation in regards to the substrate binding site now being exposed because of the cyclic AMP interaction with the regulatory subunit. And here we have the 3-dimensional structure of this particular component. We have the binding site cleft and the higher affinity association, and you'll see if we peel these 2 back, there's a high level of complementary surface interactions that gives us that high affinity and high specificity to interact with each other. So again, the regulatory subunit and the larger catalytic side has this capacity to then interact. But as you can see, the regulatory side is also potentially obscuring the sequence or the region where we're going to have the binding cleft. So we're going to have to get rid of this guy first off to then open up the binding cleft to allow the protein kinase to do its particular job. So again, it's just another illustration in regards to an orientation of protein-protein surface interactions, hiding a cleft, and in this case, causing its dissociation of the regulatory subunit will then release that cleft to do its particular job. So again, we have these images of protein structure, protein interactions, substrates coming in, causing regulation, causing conformational changes, opening up the enzyme cleft to be able to that enzyme do its particular job work. Now, as I said before, the cyclic AMP process is pretty promiscuous in regards to the types of things it can do. But what I want you to really take home here is these first parts in regards to glycogen synthesis, glucose breakdown, glycogen, so glucose is through glycolysis. Gluconeogenesis is when we're making glucose, but we also can basically utilize fatty acids, our triacylglycerols, mobilize those to go through beta oxidation to produce more energy currency. We can go through cyclic AMP and have a double balance here where we can promote glycogen synthesis with the cyclic AMP, but we can also promote glycogen breakdown with the cyclic AMP. So you can understand why we need a very close point of regulation and control. And remember, we talked about this issue in regards to overall effect in receptor activity, where there might be a positive and negative, and it's the overall constituency of the combined two reactions that give us whether we're moving forward or backwards. The same thing happens with these particular subunits too. And we come back to the phosphorylase A, kinase B subunits, these guys a little later on. But again, my issue is here, look at the individual amino acid. It's serine all the way through, and here we have some threonine there in regard to being phosphorylated by that particular system. So here's some enzymes that are going to be very important in regards to, we're going to look at pyruvate kinase, we're going to look at the pyruvate dehydrogenase complex a little later on. We're going to look at some, what else? We're going to look at glycogen synthase, we've looked a little bit at that already. We're going to look at maybe some hormone sensitive lipases too, which also increase fatty acid movement to allow us to cause their degradation to get energy. So again, we can see now how cyclic AMP becomes a problem, because if we don't control it by degrading it by those nucleotide diphosphoesterases, we're going to have this going wild. And we basically want to make sure these localized transmitters are highly controlled and regulated. Now, if you think about drinking coffee, we go back here, coffee inhibits the nucleotide diphosphoesterases activity. So caffeine inhibits this particular enzyme. Therefore, you get further stimulation because of cyclic AMP. And that's why if you drink your coffee late at night, you may not be able to get sleep, because what's happening, you're causing complete stimulation of the paracrine or hormonal system, the transmitter, cyclic AMP, around your particular body. And in this case, increasing metabolism, because when you go into a sleep cycle, towards your sleep cycle, you're slowing down your metabolism. And that's why they say you shouldn't eat just before you go to bed, because again, that's going to increase your metabolism, increase the cyclic AMP, and may give you insomnia. So again, a balance in the situation and knowing your diets and when you eat. So again, that's why we kind of have dinner at a later stage before going to bed, or you should. So when we think about the whole combination of these events, and this is one of the amazing things in regards to how a simple response can really activate the whole procedure to allow us to have a great amplification of the signal. And remember, we talked about signal amplification before. So the combination of these particular events from one stimulation has a manifold compacting event in regards to producing substrates, which will activate more enzymes and cause this case an amplificational response or a cascade in regards to doing something. And in this case, this example with the fight or flight mechanism, with the epinephrine receptor, we already know the GCPR system behind it. But let's see what happens with one molecule of epinephrine binding to that GCPR. We already understand there's going to be an activation of a adenyl cyclase and basically having a positive effect on producing cyclic AMP. So if we have 10 molecules of this particular element coming through, we are then going to produce 200 molecules of cyclic AMP. Now, those 200 molecules of cyclic AMP are going to activate 200. Well, actually not. It's going to activate a quarter of it, 50 protein kinase A's, because remember we need 4 molecules of cyclic AMP to cause its deregulation and become its activation. But then we have 100 molecules of activated pKa. So this is actually really not right. The book is a little wrong there, because we know we need 4 molecules of AMP. So with 200 molecules of cyclic AMP, we should only be reducing 50 molecules of activated pKa. But let's just keep going with that. So in this case, when we see this guy now having a positive effect with the cyclic AMP, cyclic AMP now in contention of activating the protein kinase A, that protein kinase A is going to work with a phosphorylase B kinase, which is inactivated and causes activation. And this particular enzyme is going to consume an enormous amount of molecules in regards to producing 1,000 molecules to basically now provide the glycogen phosphorylase B to be activated. So this whole system is basically activating 10,000 units of the glycogen phosphorylase A. And this glycogen phosphorylase A, 10,000 units, is going to take these massive strands of glycogen that we have stored and basically start cutting them off and producing glucose 1-phosphate in the form of 10,000 molecules of glucose 1-phosphate. We're going to dephosphorylate that, allowing it to move through the GLUT transport system, so the GLUT4 receptor again, this time in the opposite direction, and then cause the release of blood glucose of 10,000 molecules in the circulatory system. That is from the stimulation of one single molecule of epinephrine. Okay, so an epinephrine. So you need to kind of think about this cascade event that's occurring, and at every single stage, we need to have an opposite regulator. So where we see these green triangles, it means it's having a positive effect. But we also need to be able to rapidly and accurately cut off these particular systems if we need to go through and regulate and stop the system, because we don't want to cause a release of 100 million molecules of glucose. We want to stop our glycogen synthesis, our glycogen breakdown. So what we're going to do is then prohibit this, prohibit this, prohibit this, prohibit this, and ultimately prohibit the production of cyclic AMP. But as you know, we can destroy our cyclic AMP by those phosphodiesterases and stop the activity or limit the activity by having the number of cyclic AMPs being present in the protein kinase A. So again, we have all these capacities to be able to regulate and control each individual step. And there might be multiple regulators at each step. But ideally, for a rapid response, we want to be able to have a key regulator and a key capacity to stop that regulation or decrease or negative feedback very, very quickly. We do not want an event to occur here, which takes so much time to come through to get this change in the blood glucose level. We want to make sure that this is a rapid response, which it is, but we also want to make sure we have a rapid response in its shutdown. So again, keep that in mind in regards to, for every equal reaction forward, there is an opposing reaction or reactions to regulate it. Oh, and here we go. We talk about this issue with cyclic AMP and tea and coffee. Coca-Cola is the same thing. Cyclic AMP is very short-lived. And the reason it is because it is promiscuous and has a lot of activities in different tissues and different receptors and different enzymes. And when we take the nucleotide diphosphoesterases and bring it down to the non-cyclic form, the adenosine 5 monophosphate, this is not active. Okay, this is not active anymore. And as we said, the tea and coffee inhibit the conversion and therefore increase the circulatory lifetime of the cyclic AMP. So it increases its bioavailability and it's what we call circulatory half-life. So in the normal situation, this half-life for this particular molecule might be only a few milliseconds. But as you add coffee and tea to the component, we are going to see then the continual stimulation of this particular molecule being produced and therefore its concentration increasing. So it increases both its circulatory half-life and its existence within the cell. And again, we know this occurs because of caffeine in regards to tea or coffee inhibits the degradation of the cyclic AMP to make adenosine 5 monophosphate or AMP. So we've got some terminology we want to go through. The main thing is to understand some of the mechanisms of biosignaling, the issue of Scatchard plots and looking at receptor density, because this becomes very important. Because remember, and we had a little discussion, someone brought a very good comment in regards to, well, how do we regulate the receptors? That is in the next part of the lecture series in a second. We're going to talk about membrane ion currents and the acetylcholine receptor. And then we talk specifically about cyclic AMP and GMP and secondary messengers and the GCPRs. You need to understand what GCPRs do, how they work in regards to their cyclic AMP degradation and activation of protein kinase A and the cascade events that may occur in regards to that simple phosphorylation event. As we're seeing now a biological interaction, three-dimensional structural protein change, interaction with phosphorylation, dephosphorylation to cause regulation, activation of an enzyme activation to cause an effect. So again, basically like sometimes they call it a Z diagram in regards to what occurs. And then importantly, this amplification response. We are going to see a lot more of these amplificational responses occurring when we look at metabolism in regards to specific metabolic pathways. And I'm hoping that we'll get to tomorrow to glycolysis and gluconeogenesis, which is going to be our first major metabolic pathway. So let's continue our lecture on now